Hello everyone, I'm Charles Mulligan, host of today's Science of Childhood Cancer seminar series. Today's speaker, Paul Thomas, will speak for almost an hour and then take questions before the top of the hour. He's also agreed to stay on for additional time after that, so if you'd like to stay on afterwards and be engaged in a moderated discussion and Q&A session with him, please do so. So with that, I'm very glad to welcome Paul Thomas as the speaker in the 13th talk in the 15-talk series of the Science of Childhood Cancer. When we were planning topics targeting precision medicine and pediatric cancer, I was very glad that Paul was willing to participate. So Paul is a card-carrying immunologist. He trained at Harvard and then undertook postdoctoral training at St. Jude and has been on faculty since 2009. Paul's made a number of seminal discoveries in the field of viral infections and the immune responses to viral infections, the nature of the T-cell repertoire, down to single-cell resolution and mapping its clonality and progression and responses to various disease states. And relevant to today's talk in the seminar series, Paul has also conducted groundbreaking research profiling the nature of the T-cell response to a number of tumour types and tumour states and mapping the tumour microenvironment, understanding the nature of antigen and neoantigen presentation by tumour cells across a range of pediatric cancers, liquid tumours as well as solid tumours, and showing some fascinating insights into how these antigens can be potential targets for immunotherapy. So with that, thank you again, Paul, for participating today, and over to you.
Thanks, Charles. I appreciate the introduction, and I really appreciate the chance to speak in this series. I've been watching it from the beginning, and the talks have been really extraordinary. So today I'm going to present on sort of two areas of work in the lab, trying to understand the interaction of immune cells with tumors and characterize these sort of two broad ways in which we think immune cells interact with the formation and destruction of tumors. And so in this first half of the talk, I'm going to be talking about the sort of conventional way in which, especially recently, we've been thinking about immune responses and how they might work therapeutically to reduce or eliminate tumor burdens. And a lot of the focus in my lab and in the field has been around getting T cells, effector cells of the immune response, to target cancer cells. And to do that, we need the T cells to recognize the tumor cells. And they do that by recognizing tumor-specific or tumor-associated antigens. The tumor-specific antigens seem to have the best therapeutic profile because they should only be in tumor cells and so limit off-target effects of any type of immunotherapy. And so these neoantigens are generated through genetic mutations, either indels or SNPs in the tumor genome that get processed and presented on MHC class 1 or class 2 on the tumor cell itself, and then recognized by a T cell with a specific T cell receptor that can distinguish that neoantigenic peptide. And the T cell receptor itself, as Charles mentioned, is a major focus of my lab, and understanding the diversity and repertoire of the T cell receptors that make up an immune response has been very useful for understanding the efficiency and ultimate outcome of antiviral and other antipathogen immune responses. And so just a brief reminder to everybody about how the T cell receptor is generated. It's through this complex process called VDJ recombination, which utilizes cassettes of genes on the two chains of the T cell receptor and conventional T cells, which are alpha-beta T cells. And so you have a beta chain and an alpha chain. And you have these cassettes of variable genes, joining genes, and on the beta chain, you have these diversity genes. And in each developing T cell, one of these segments from each of these cassettes is chosen and fused together in a semi-random process that is purposefully error-prone and involves the insertion and deletion of a number of nucleotides at the core fusion junction, which creates an enormous potential diversity of T cell receptors. And then when the T cell receptor binds to MHC and visualizes the peptide, that hypervariable region where the key fusions have occurred, which we refer to as the CDR3, are primarily responsible for recognizing the peptide, and in the case of tumors, potentially distinguishing a self-peptide from a tumor-specific peptide. And I'm going to come back to the concept of T cell repertoire and the T cell receptor as a specificity determinant and as a way of understanding the underlying clonal architecture of a T cell population throughout the talk. And so one thing to keep in mind is that there's an enormous potential diversity of T cell receptors that can be generated by this mechanism. And so on the low end, the estimate is around 10 to the 20, which is about 8 to 9 orders of magnitude greater than the total number of T cells that you'll generate in your entire lifetime. And an actual more accurate count that's more recent is closer to 10 to the 60th potential T cell receptors. This space is not explored completely non-randomly, and so there are T cell receptors that recur between individuals, and so it's not a truly random process. But there is an enormous diversity, the highest sort of level of diversity of any process that can be introduced into the human body. So we're going to apply some of these concepts to looking at T cell responses to tumors, and particularly with me being here at St. Jude, we're looking at pediatric tumors. And we were starting in the context of the explosion of immunotherapy work that had happened in the last 10 to 15 years that had primarily been focused on adult tumors, and really as a result of the groundbreaking work of Jim Allison and others in checkpoint blockade immunotherapy. And what had been observed in checkpoint blockade immunotherapy is that if you look at the kind of classic chart of tumor mutation burden, and so this is a log scale here, across different tumor types, checkpoint blockade therapy was working primarily in these very high mutation burden tumors that are characterized by being mostly adult tumors and high mutation because they developed after an accumulated lifetime of carcinogenic hits. And so lung cancer, particularly lung cancer and smoking-associated lung cancers, melanomas, and so forth. And a hypothesis has been emerging that these high mutation burden tumors are responding well to checkpoint blockade as a result of the fact that only a small proportion of mutations generate detectable T cell responses. And there's a lot of theories about why only a small proportion of mutations generate these T cell responses endogenously in people carrying these tumors. But one of the more popular ones is that the average tumor mutation, being a single point mutation, doesn't generate a peptide that can be distinguished from self easily enough to generate a robust immune response. And also in a lot of tumors, there are not really good evolutionarily conserved public epitopes, meaning that from tumor to tumor, you don't get that many repeat mutations. There are obviously some very important exceptions, and we'll talk about those throughout the talk. And so the general consensus of the field up until I'd say recently has been that high mutation tumors are good responders because low mutation tumors are unlikely to generate targetable T cell responses. Another way of saying all that is that there must be a lot of holes in the anti-tumor repertoire. So even if you have a large number of targets, you're only going to target a subset of them. And some raw data to sort of make these points. These are from some older papers, 2015, 2016, of characterizing melanoma patients from Steve Rosenberg and Ton Schumacher. Each line here is a different patient. They sequenced the number of non-synonymous mutations in these tumors, and then they tested the patient's own cells at the time of diagnosis for a tumor response. And so this was pre-checkpoint blockade therapy, administration of ICB therapy. And so in this middle column is the number of potential neoantigenic peptides that were screened, either by making tetramer reagents or functional assays or expansion of TIL lines. And so in the Rosenberg papers, anywhere from 16 to 48 peptides. And in the Schumacher paper, they went up into the hundreds of different targets. And then these are the numbers of endogenous responses that were detectable, that were already present in the patient at the time of diagnosis, that had been mounted, that would presumably be the target of a checkpoint blockade therapy. And what you can see is that on the very highest end in the Rosenberg paper, they found 3 HLA-A2 restricted T cell responses out of a total of 32 peptides that were screened. And on the low end, out of 462 peptides, they found 0 responses targeting any of those tumor mutations that were mounted endogenously in the patients. And if you average together a large number of studies, what we call the hit rate is low. So the number of mutations that are converted into a T cell response that you can characterize and detect either in the tumor or in the periphery of a patient with a tumor is only about 2% across multiple studies. So any given mutation is maybe only a 1 in 50 chance to convert into a T cell response. And if that's purely a result of chance, of lottery tickets, of recruiting something from the repertoire, then a pediatric tumor that has only 2 to 10 mutations like ALL should on average never generate an endogenous T cell response. So when we started approaching this, we came at it from a little bit different perspective because of the background in viral immunology. And in particular, in viral immunology, and to be fair, in tumor immunology from a number of investigators, there's a robust concept of immunodominance in T cell responses. And the concept of immunodominance is basically that if you think about a virus, even a small virus, relatively small virus like influenza, you have potentially hundreds to thousands of peptides that could be generated from that virus, all of which would not be similar to self. They would be very far away in antigenic space because they're all virally derived. And many of these would be able to bind MHC or HLA very effectively. But when you actually look at the number of responses that are converted into T cell responses that are mounted and that we can measure, it's usually a very, very small proportion of those potential epitopes. And so in the classic mouse model of influenza that we study, out of several hundred potential MHC binding peptides, over 80% of the entire CD8 T cell response in the animal is targeted against just 5 epitopes from that virus. And this was it's still an ongoing area of research in a number of labs, but in 15 to 20 years ago, it was sort of just being described rigorously because of the advance of various T cell technologies at that time. And so this is an older paper from my postdoc here, where we were looking at why these 5 peptides are so immunodominant. And so the question was, what's really being optimized here? Is it really just that these 5 peptides are the only peptides that the immune response can see, or is there something, just a set point level of a response that's generated, but that once that's generated, other peptides are not used to elicit a response, but could be in the context of these peptides being missing. And so to address that, the flu model is quite tractable. We looked at what the wild type influenza response looks like in these animals. And so this is in the red bars here. And these are those top 5 peptides that make up 80% of the response here. And we're just referring to them by their gene names in the influenza virus. And then we discovered some extremely minor T cell targets here. And so what we did then is we engineered the flu virus to not present these top 2 epitopes. And so this single epitope here accounts for about 50 to 60% of the entire T cell response in the lung during the flu infection in this model. And so we just mutated an anchor residue in the peptide here so that it cannot be presented as an epitope on the mouse MHC, and then generated those viruses, and they were equally as fit as the wild type viruses. And then we looked to see what happened to the immune response. And the question was, if these are the only peptides that the immune response can see, then we should really cripple the immune response because we're taking out its best targets. But if it's really just sort of a set point response, then the immune response should be able to compensate by making better responses to some of these other targets. And that's exactly what happened. So we lost the response to these 2 immunodominant peptides, and we just expanded the response to these very minor peptides, some of which were in the wild type situation really at the limit of detection. And so this seemed to fit the sort of tumor model particularly well, where we were thinking that it wasn't that the tumor neoantigens in these high mutation burden tumors were all poor antigens, and that's why you weren't being able to detect a response. It's just that there were so many of them that finding the small immunodominant subset is extremely difficult, because we still don't fully understand all the rules of immunodominant selection. We can talk about that in the discussion a bit more if people are interested. But so our approach to this was to look on the other end of the mutation burden spectrum and start from the very lowest mutation burden tumors and ask whether or not a larger proportion of those epitopes are being targeted by the immune response relative to the high mutation burden tumors. And so we did that in ALL first, and I'm going to show you that data, and a lot of it is published, and so I'll go relatively quickly through it. And this was working with a computational pipeline to work from the genome sequencing that's performed here at St. Jude on patients, and then curate mutations and predict potential neoepitopes based on the patient's HLA, and then generate the reagents that we need to characterize these T cell responses. And this is work that was initiated by Jingwei Zhang, the Department of Computational Biology years ago, and a pipeline that that we use by Jeremy Crawford, a computational biologist in my group. And so we have patients, and we curate, like in this patient, 2 mutations that we then look at on their HLA haplotype to predict whether or not they would be epitopes, and then we test those. And this is the functional work here is being done by Anthony Zamora, who's a former postdoc in the lab, who's now a faculty member at MCW. And he used a number of different functional assays that are in the paper, but I'm just going to show you a couple to show you the key results. And so what we've done is we've taken T cells out of the diagnostic bone marrow from these patients. So none of the patients have received treatment previously. And these are the very small number of T cells that are in the blast-ridden bone marrow. And so this is looking at tetramer assays to look at tumor mutation-specific T cell responses in the bone marrow at the time of diagnosis. And so we're gating on CD8 T cells, and then we generated tetramer reagents, which are peptide MHC tetramers that can pull out T cells specific for mutation. And this is flow cytometry staining. And so this is a background stain of an irrelevant tetramer that's matched to the patient's HLA. And then we made a tetramer, so a peptide MHC complex containing the self peptide, the unmutated peptide, to look for cross-reactivity back to self and whether or not the T cells that we're pulling out that are specific for these mutations can distinguish between the mutant peptide and the self peptide. You can see that background is still quite low, though maybe a bit higher than the irrelevant tetramer. And then when we use the actual mutant peptide that's characterized from that patient's tumor that we predicted would bind to that patient's HLA, in this case, this is one patient, this is a second patient, and for 2 different epitopes, we had very nice responses, 8 to 12%, 5 to 8% in this other patient against these predicted neoepitopes. And again, they were always much more specific for the neoepitope than the self epitope. In collecting a lot of that data together, this is for 6 patients now. All patients showed at least 1 response, and most had multiple responses with evidence of these within host immunodominance hierarchies, where you've got some responses that are really quite large and some responses that are above the limit of detection but are really quite small. And so it looks a lot like an antiviral conventional CD8 T cell response, but we're recognizing a much higher hit rate than what's observed in these very high mutation burden tumors. The black bars here are all controls, either irrelevant tetramers or the self epitope. If we add up all of the cells that we can account for in the bone marrow at the time of diagnosis, all the CD8 T cells that we think are specific for tumor mutations, and just plot that on the x-axis here, and then plot it against on the y-axis the total number of CD8 T cells in the tumor, there's this very nice positive correlation, suggesting that at least a substantial portion of the T cells that are in the bone marrow at the time of diagnosis are targeting the tumor. And so all of these were targeting, or many of these were targeting single point mutations that are generally private between patients. They're not shared epitopes. And so as a therapeutic pipeline would not potentially be practical with our current technology. But as many people in the audience, I'm sure, are aware, a lot of childhood ALL is driven by relatively conserved fusion events. With the most common being the TEL-AML1 fusion or ETV6-RUNX1, 1221 fusion here. And so here we wanted to see if whether or not patients were mounting good conserved responses against these conserved antigens, with the hope that potentially this could be an off-the-shelf therapeutic. And so this is what potential epitopes look like for the ETV6-RUNX1 fusion. And so this is looking in different HLAs. So throughout the study, we were relatively agnostic to HLA type and really worked to try to look across a really broad diversity of HLAs. And everything I'm showing you was consistent across most HLAs. And so this is the ETV6-RUNX1 fusion, ETV6 in red, RUNX1 in blue. And you can see across the different patients here, the fusion junction is relatively conserved. There are occasionally variants, and often the variants are even within a patient, and that comes from alternate splicing, particularly within RUNX1. And you can see many potential predicted epitopes, but predicted epitopes often do not convert into actual responses. But when we looked in these 5 additional patients, so these are different patients from the ones I showed you before, every single patient mounted a response against the fusion neoantigen. And some of these were really strongly immunodominant. So in the ETV96 and ETV98, over 20% of the T cells, or approximately 20% of the T cells were specific for the fusion. neoantigen. And these, interestingly, were A0201 patients, and an A0201 epitope, and A0201 is a very common MHC, which has good profiles for population-based therapeutics. And as I said, most of this work is published, and so the reference is there. And so these conserved fusion antigens are, we think, really potential off-the-shelf targets for cell therapy. So one question that you might have then is why these patients, if they're able to mount such a good immune response, or at least unimmune response, why, one, do they still form tumors? And two, it's generally well accepted now that ALL, pediatric ALL, is not a good setting for immune checkpoint blockade. And so we wanted to functionally characterize these cells in a bit more depth. And so here we're sorting on tumor-specific T cells and doing single-cell transcriptomics to look for what's happening with these cells in the context of the bone marrow tumor. And so in this slide here, every big grouping in the middle, the red, the yellow, and the green, are a different patient. And then every narrow column is an individual cell from that patient that we're characterizing now for its functional response. And every cell is tumor-specific for that patient. So it's sorted on tetramer to get the transcriptional profile. And I've clustered the responses over here in sort of functional subgroups, with this green group being the sort of classic T-cell effector group, and the red group up here being, if you follow the adult checkpoint blockade literature, the sort of T-cell exhaustion phenotypic correlate markers. And you can see that most of the patients don't have a really high abundance of, or most of the cells don't really have a traditionally exhausted phenotype. This patient had a portion of cells that were exhausted, but not all. Many of the patients have very nice effector profiles. And interestingly, this patient in particular, but all of the patients had this profile associated with type 1 interferon signaling, which has been reported both in animal models and in humans in leukemia to be a dysfunctional sort of T-cell signaling profile. And so they don't seem to have the type of profile that's easily targeted by checkpoint blockade. If anything, they seem to be generally good effectors, or potentially a dysfunctional effector profile that's just inefficient at clearing the tumor, but not terminally exhausted in the sense that you see in these more slow-growing, slow accumulating adult high mutation burden tumors. And so this could be why you still mount a good response, but checkpoint blockade is not the way to juice that response up further. And so this interferon profile is interesting to us just because it's appearing in this blood tumor, and it's, as I said, it's been reported before, or hematopoietic tumor, and it's been reported before as potentially not an efficient effector response. And in parallel, we're working on some new studies that are unpublished in collaboration with Amy Tallur and Stephen Gottschalk in bone marrow transplant here, characterizing their CD19 CAR trial in St. Jude patients. And this is a collaboration with the Immune Monitoring Corps over in BMTCT and headed by Deanna Langfit. And so this is a traditional CAR therapy where we can get samples longitudinally across therapy, starting with patient samples prior to product generation. And then we can analyze the product after CAR transduction. And then we can analyze the cells after they go back into the patient and hopefully expand and control the tumor. And so we've been doing that using the T-cell receptor that's endogenous on the T-cell as a barcode for the lineage of these CAR lineages. And one of the questions we've been asking is, if we look in the GMP product, the product of the transduced T cells that's going to go into the patient, can we predict which cells are going to expand and which cells are not going to expand or even survive in the patient? And so using the T cell as a barcode, we can track these lineages across multiple time points prior to TCR or CAR generation and after infusion into the patient. And this just shows all of the lineages we're able to trace for one patient. And it gives us an enormous amount of statistical power to discern what's happening in these cells that makes them fit or not fit, which we've been characterizing by single cell transcriptomics. And so this just shows a blow up of some specific lineages that are persistent over time that we can see at multiple time points within the patient, though we don't always see them at every time point within the patient. And we can build machine learning classifiers that define the gene expression profiles of lineages that persist versus lineages that fail to expand. And so we have hundreds to thousands of these across multiple patients. And in our preliminary data set, we've built classifiers that have really nice AUC curves And the most striking feature of lineages that we see as abundant in the product that had every chance of appearing in the patient, that then do not ever appear again in the patient, is this in the product gene expression profile characterized by intense type 1 interferon signaling. And so that seemed consistent with what we had seen before in a very different model of an ALL effector response, but potentially this is a common feature of responses that are inefficient against ALL targets. And then just one more word on hematopoietic targets. Our current research in this area is focused on AML, which obviously has much worse outcomes than the ALLs that we have been looking at. And there are similar to not as penetrant as the ETB6-RUNX1 fusion in ALL, but still relatively high frequency fusions like core binding factor in AML, some of which have that same profile in terms of at least our in silico predictions of being robust HLA binders that will be good targets for endogenous T cell repertoires. And so we're actively fishing out TCRs for these fusions now, and hopefully we'll have an update on that soon. So when we first had been developing this work, the question was, how much of this is, an artifact of just this one system, or how much of it is potentially expandable to other low mutation burden tumors, and maybe, changing the paradigm of high mutation burden tumors being the only ones that elicit T cell responses. And so switching to a different tumor type, we've also become recently interested in, again, because of its fusion-based characteristics, in fibrolamellar carcinoma, which is a relatively rare liver tumor, but it affects adolescents and young adults, and has very poor overall survival, particularly if the initial resection fails to completely remove the tumor. And it's interesting to us because it was mediated in virtually 100% of patients by exactly the same fusion, which is this DNA JB1 PRKACA gene fusion, which is within chromosome fusion, a large deletion that fuses exon 1 here to exon 2 DNA JB1. And this work's being done by Alison Kirk in the lab as a graduate student. So we've acquired a number of samples from patients with FLC. And the very first patient in collaboration with Scott Strom at UT that we got access to, Scott's lab had generated T cell lines to look at the response to this tumor. So these were endogenous cells directly out of the tumor of this patient that were just expanded in the presence of fragments from this patient. So not particularly expanded in response to the fusion. And so we ran a single cell genomics looking at the functional responses of these cells in response to pieces of the fusion that we fed into the cultures. And we identified clusters of T cells that shared T cell receptors. They weren't the dominant T cell receptor in the tumor. They were actually in the TILs. They were actually a relatively small proportion of the TILs, but that seemed to be responsive to a fusion peptide. And we've done this now across multiple tumors, but I'm just going to show you this one example. And so we pulled these T cells out. They actually all shared exactly the same TCR. So we got one TCR that was responsive out of this patient's TIL lines. And we cloned it and re-expressed it and then treated it with the peptide that we predicted would be derived from the fusion. And it's this peptide here that spans the fusion by only a single amino acid, this valine, into the protein kinase A coding region, which is a glycine in the wild type DNA JB1 gene. And so looking at a monoclonal T cell line that's expressing this T cell receptor and titrating down these peptides, looking at functional responses by interferon gamma production, CD69 upregulation, or TCR alpha beta downregulation, we can see that the mutant peptide is always better than the wild type peptide. But unlike some of the studies I showed you for the ALL peptides, we actually did see a fairly reasonable response to the wild type peptide here, which was interesting to us. We also mapped the restriction of this peptide to A6802 in this patient, so that HLA type. And when we explored this a little bit further, this is in a culture system where we're adding the peptide at these defined concentrations here. We looked at the ability of these two peptides to bind to A6802, and the mutant peptide had a much lower predicted binding affinity, so much better binding affinity for A6802. This affinity here is usually considered, essentially negative. Like this peptide's not going to be able to bind in a natural setting. And indeed, if you look at the motif of peptides that combine to A6802, valine is a key end anchor residue. And so the mutation here of the fusion, bringing a valine into this coding sequence here, actually generates this peptide that can now bind to HLA, whereas this peptide, while antigenic in the sense of these residues in the middle being able to be visualized by TCR, just can't efficiently bind, we think, can't officially bind to HLA, and so in a wild type cell is not efficiently presented, and so doesn't generate an anti-self response or anti-self tolerance. And so this is kind of an interesting way for a fusion peptide to almost mimic a single nucleotide variant. So what I've shown you in this first half is that immunodominance likely biases our ability to detect responses against highly mutated tumors. And in the low mutation burden tumors that we've looked at thus far, there's really a large pool of tumor-infiltrating neoepitope-specific CD8 T cells that are responsive. This is in pediatric liquid. I've shown you an example from solid tumors. And it's an important point to say that response to checkpoint blockade, which has generally been very poor in pediatrics, is not the same thing as tumor immunogenicity. So the fact that pediatrics patients tend to fail checkpoint blockade does not mean that their immune responses aren't trying to target the tumor. And so we're looking at ways to increase the magnitude and function of these cells, which is essentially what a CAR T cell therapy is in a way. It's an added specificity, but it's also a much higher precursor frequency and a highly functional cell population. And I haven't shown you too much data on this, but we have some additional data that these elicited TCR clonotypes are highly neoantigen specific with no cell cross-reactivity. I showed you that with the ALL data. And so that gives us a lot of opportunity for TCR-based therapy. And these recurrent fusion antigens are particularly attractive to us because they're consistent targets of the endogenous immune response. And so it's possible that really many tumors are immunogenic, but with limiting responding repertoires. I'll move on into the second part of the talk here. So the first part of the talk was really focused on how the immune response visualizes the tumor and can work to destroy it. But in the second part of the talk, I want to show you some new data from the lab, really trying to think about how the immune response is contributing to tumor formation and how that also opens up distinct and maybe sort of unconventional therapeutic opportunities. And so this work has come out of a number of streams of research in the lab, but it again goes back to the work that Charles mentioned at the beginning, where we've been working hard to try to understand the nature of T-cell repertoire landscapes. And so in the service of that, we've developed a number of tools for quantifying features of the T-cell receptor itself. And I promise the talk's not going to get too detailed about T-cell receptor biology, but I just want to introduce the concept of T-cell receptor similarity as being important for understanding immune responses to pathogens or tumors. And so we developed this algorithm that we call TCR DIST, which is basically just a similarity measure of T-cell receptors, any two T-cell receptors. So, and it's weighted based on the key portions of the T-cell receptor that make contact with the antigen, the peptide MHC. And so this is a T-cell receptor structure over here, and this is all the surface of the T-cell receptor that's going to visualize the peptide MHC. And then this is a different T-cell receptor here, and we've just developed a way of scoring the differences between those two surfaces, overweighting that part of the T-cell receptor that makes the most important contacts with the peptides. And so we can sum over this, over all of these differences, and we get a single number that we call the TCR DIST, which is the higher the number, the more dissimilar the two T-cell receptors are, and the lower the number, the more similar they are. And what we've shown in this paper and in a number of subsequent studies is that the closer T-cell receptors are to each other in this TCR space, the more likely they are to see the same antigen. And we can put relatively rigorous statistics on the likelihood of two T-cell receptors seeing the same antigen. And so this is useful for us in the context of tumors now, because we have a sort of antigen agnostic way of looking at the landscape of tumor responsiveness. And so one way that we're applying this is to take just bulk sequencing from tumors and from normal tissue in the same patients, and looking at the, using next generation sequencing, all of the T-cell receptors that are present in a tumor tissue versus an adjacent normal tissue, and looking for enrichment clusters based on this TCR distance of T cells that are shared and that are attacking the same target inside of the tumor. And so this clustering in TCR space gives us a lot of statistical power to then identify a cluster of T cell receptors that are more similar to each other than you would expect by random or that you would see in normal tissue, and that we would expect then are targeting things within the tumor tissue. And so this is another way of getting at what are the relevant tumor antigens by, rather than starting with the antigens, starting with the T cell receptors themselves. And this is some published data from this paper here, where we identified a number of what we assume are which are tumor-associated clones that we assume are potentially tumor-specific clones. And we can then look within those clones and identify specific motifs within the T cell receptor. So this is the V regions those T cell receptors use, the J regions, and then that CDR3 region, that's the really important contact with the peptide. And we can do statistics on whether or not that motif is overrepresented in particular tissues and metastases within that patient. And then potentially also for these shared antigens where there's these public fusions that are potentially targets, we can look for shared motifs that would likely be targeting these public antigens. We've taken this concept a little bit further now, and particularly with the advent of single-cell technologies and single-cell multi-omic technologies, where we can get the TCR, we can get the gene expression profile, and we can get epigenetic profiles from a single cell at once from tumors. We can build much more robust classifiers to identify interesting subpopulations. And so this paper is currently in revision, but is already on the bioRxiv, and the code is available on GitHub for a new method for relating clonal relationships in T cell receptor space that I just told you about, looking at clusters of T cells that we think must be seeing the same thing and clusters of T cells that share interesting gene expression profiles, and using that to profile specific subgroups that are probably functionally relevant in the context of anti-tumor responses. And so this is an example of that we've done with the FLC tumors that I was showing you about in the previous section, where we take, this is now not T cell lines, these are directly ex vivo from the tumor, but re-stimulated with large peptides spanning the fusion that's driving that tumor, that DNA JB1 PRKACA. And then we apply this algorithm that we call CONGA. And CONGA stands for clonotype neighbor graph analysis. And the reason for that is we think about it in this way. So you can think about all of the T cell receptors that you find in a particular tumor. So we take all of the T cell receptors that we find in the tumor, and we use that TCR distance score I told you about, and we just project those T cells into a 2D space, just like you would for gene expression profiles. So you can plot a t-SNE, you can plot a UMAP, but the relationship in this 2D space represents the relationship for how close these T cell receptors are to each other. And so in this space, these T cells have neighbors, and you can draw little graphs around the T cells to say who are their nearest neighbors. You can have little connection nodes and edges that each T cell has a certain number of neighbors, and you know which ones are its closest. And then you can take exactly those same cells and plot them in gene expression space, the way everybody's doing with 10x data, with t-SNE or UMAP, and you draw the same graph now, but with the same cells, but their relationships based on gene expression. And then you look for statistically surprising overlaps between neighbors that have the same gene expression and the same, or not the same, but similar TCR. So the nice thing about this approach is that it doesn't rely on identity, it relies on similarity in this kind of abstract space. And what we've discovered is that this actually identifies a lot of clusters that we wouldn't find by using either method alone. And so this is just from this patient. These are the number of unique clusters that we found. And again, we wouldn't have found all of these with just looking at TCR clustering or just looking at gene expression clustering, or even looking at the union of the two. It's really having to look at the relationship between the two that really pulls it out. And I'm not going to spend too much time on all of the details of the output, but it's a very powerful tool for looking at many aspects of the underlying T cell biology. But one of the things that we noticed in this directly ex vivo of this liver tumor was, once we saw it, perhaps not super surprising, but there's this cluster of interesting specificity groups here on these bottom three groups. And so these are groups that share neighbors in TCR space and in gene expression space. And these are the genes that they share. These are the TCR motifs that they share. If you're a TCR person, this is the TCR alpha and then this is the TCR beta. When you see this motif, you immediately know that these cells are mate cells, which are an invariant type of T cell that's quite common in humans, enriched in the liver, but we find even further enriched in these liver tumors. And this is interesting to us because we hadn't really thought of these tissue-specific invariant T cell populations that are highly regulatory as playing potentially significant roles in the context of some of the tumors that we were looking at. Using this combined profiling, this really came out at us. And for the rest of the talk, I'm going to be talking about how we've stumbled on this principle, we think now, in a number of different settings. And so I just want to get everybody on the same page about what we're talking about. So everything we've talked about so far in terms of immunotherapy and trying to get T cells to target the tumors are these conventional classic T cells, where you've got a T cell receptor and you're hopefully seeing a foreign antigen or a neoantigen in the context of host MHC. But a huge portion of your T cell repertoire is actually not those T cells. It's these unconventional T cells, things like gamma delta T cells, which would use an entirely different T cell receptor, or these more invariant populations like NKTs, the mate cells that I just mentioned that respond to B vitamin metabolites, Tregs, which have features of conventional T cells, but also features of more consistent with these unconventional T cells, and IELs, which are intraepithelial lymphocytes, which are really important for tissue integrity and tissue formation. We've been starting to think about these cell types as potentially playing a role in tumors. And I'm going to show you one story now in the closing slides on how we think they may be really critical to tumor formation and tumor maintenance. In this part of the talk, I'm going to be talking about a neuroblastoma model that we have in the lab that was generated by Peter Murray, who's now at the Max Planck Institute in Germany, and a staff member in his lab at the time, Leanne Vandeveld, who's now in my lab. And it's great that I'm talking this week. If you saw last week's beautiful talk from Rami George, who is the inspiration for a key feature of this model, whose work was really critical for developing some of the thinking behind this. Again, if you saw last week's talk, you're aware that neuroblastoma is the most common extracranial solid tumor of childhood. It's characterized very nutrient-poor microenvironment. And I just want to mention that because it'll be relevant later. And what Peter was particularly interested in was modeling one of the high-risk forms of neuroblastoma, the common high-risk form of MYC amplification, which had been previously modeled by others using a tyrosine hydroxylase MYC mouse model. What Peter found, though, was that the tumor burden in these mice, at least in our animal facility, was not particularly penetrant. And so based on the work of Rami George and others that had mapped a number of mutations, including some of the work from here in the PCGP, he engineered in using CRISPR single alleles of ALK gain-of-function mutations, including the very common F1174 mutation that we heard about last week and the Y1282 mutation. And so this is in the THMYC background, and that F1178 mutation, 1174 in human is much more penetrant than the Y1282, and both much more penetrant than the parent THMYC only line. And so we treat this as sort of a clinical trial with each of these mice. The mice are imaged weekly, and we pull mice out for humane endpoints if the tumor reaches too big of a size, or we close them out of the study if they do not develop a tumor, and that's what these curves are based on over 7 consecutive imaging sessions, or if other animal welfare issues arise. And you can see we get very nice, very large tumors, particularly in the adrenal area. And when we characterize these tumors genetically, they cluster very nicely with MYC-amplified human tumors in this group here on the right, and cluster very distinctly from actually MYC alone tumors in the mouse and in other human tumor types. So we've been characterizing this model, sort of as an independent team of study in the lab. And we were, again, as immunologists, particularly interested in looking at the hematopoietic compartment and how it was contributing to thought might be tumor control. And so we were thinking this project would proceed along the lines of some of our other projects, where we're looking for targets within the tumor that are being visualized by the immune response to eliminate the tumor that we could use therapeutically. And so to characterize all of the immune cells that were in the tumor, we use this trick where you IV inject a CD45 antibody into a mouse right before harvesting the tumors out of the mice. And so that all of the cells that are actually in the tumor parenchyma will not be labeled by this antibody. And so we can sort out those cells that are in the parenchyma versus cells that are just passing through in a blood vessel. And so then we perform 10x on the CD45 positive cells within the tumor, but that were CD45 negative based on this retroorbital staining. And we also included some tumor cells as well, just to get some gene expression information on those. So mixed them together and ran single cell RNA-seq. And this is from multiple animals pooled together. And what was interesting to us is that we actually had really robust amounts of parenchymal adaptive immune cells in what's generally considered to be a relatively cold tumor. And so we had a really large group of B cells, CD4 cells, CD8 T cells, and then various macrophage subgroups. This profile is actually not so different than what you would see in a lymph node or in another, immune organ. And that's why it was really critical that we distinguish between the blood and the parenchyma of the tumor, just so that we knew that these were really inside of the tumor. And so the first question we had when we saw this massive adaptive localization was, what's the role of adaptive immunity in restraining tumor growth? And so we thought, just like in all of these other models we've been looking at, the adaptive immune response is actually trying to get rid of the tumor and is maybe being stymied for one reason or another. And so the blunt hammer that we used first to try to see what the role of adaptive immunity in the tumor was to breed these animals onto a RAG knockout mouse. And so this is the F1178 THMCN mouse bred onto RAG. So these mice will have no T cells or B cells. They have no adaptive immune compartment. And we thought we were going to get much higher penetrance of the tumor, much more aggressive tumors, larger tumors. And we got the exact opposite. So we have never actually gotten a tumor to grow in these animals. So there's 0 incidence, 100% survival. We cannot get a tumor in these RAG knockout mice. So rather than playing any kind of limiting role, lymphocytes, either B or T cell lymphocytes, are playing a pro-tumorigenic role, not limiting tumors, but somehow growing them. And so the question was, what lymphocytes and why, and what are they doing? And so to really get at this, we turned towards spatial transcriptomics to try to understand the architecture of the adaptive immune response in these tumors. And here we're going to combine our spatial transcriptomic data with our true single cell data. But spatial transcriptomics, for those of you that are not familiar, is we can take sections of tumors. So we took 4 different tumor sections from multiple tumors. You put them on slides that you can then image conventionally. And then there are barcodes underneath the tissue on the slide that you can elute the RNA onto and get spatial resolution, not single cell, but about 50 micron resolution of the RNA within the tumor, and relate that back to the tumor single cell data that we have from conventional single cell RNA-seq. And so this, just to remind you, is the composition of the tumor by single cell. And so we have this large group of B cells, but when we look at where B cells were in a representative tissue section, they weren't really innervated in the tumor, and they were really on these border areas. So there were a lot of B cells, but they weren't really in the core of the tumor. And if we depleted all of the B cells in the mice, starting from weaning, so before tumors have formed, we didn't change tumor formation at all. So B cells seem to be, while they're there, they seem to be irrelevant, at least to tumor formation and maintenance. So then we went to our favorite cell type, the CD8 T cell, that we thought, originally starting this project, we thought would really be important for limiting tumor growth. And there are a lot of CD8 T cells in the tumor. But CD8 T cells have to see antigen in the context of peptide MHC. And so we looked for MHC class 1 in the tumor, and that's stained here. So the MHC on the y-axis, and CD45 on the x-axis. And so CD45 negative cells here are going to be tumor cells, and they were uniformly MHC negative, and that's just quantified here. And so there's no target then on these cells for CD8 T cells to see. So they can't kill them if they don't express MHC class 1. And indeed, like the B cells, the CD8 T cells were really in these border regions and were not going into the tumor. And if we depleted the CD8 T cells, there was, if anything, a slight trend towards actually fewer tumors rather than more tumors, which is if the CD8 T cells were getting rid of the tumor, what we would have expected. But it wasn't statistically significant. And just by way of comparison, looking at an array of human tumor types just at bulk RNA-seq level, looking for beta-2 microglobulin, a key component of the class 1 antigen presentation complex, it's expressed at extremely low levels in neuroblastoma, really an outlier level compared to other tumors. And so we think this is consistent with the idea that neuroblastomas may be bad targets for CD8 T cells because they don't actually present CD8 T cells, MHC class 1. Okay, so we've gotten rid of B cells and it didn't do anything. We've gotten rid of CD8 T cells and it didn't do anything. And so we're left with CD4 T cells. By the spatial transcriptomics, CD4 T cells looked much more interesting. They're innervated throughout the tumor. And when we deplete CD4 T cells, we find that they're necessary for tumor formation. And furthermore, when we look at these few tumors that we do get in the CD4 T cell and the CD4 depleted tumors, we find an interesting thing. So we were checking this just to make sure that we had actually depleted the CD4 T cells in these mice where they still had tumors. And indeed, we had depleted the CD4 T cells, so they were all gone. But there was an increased number of CD8 T cells in the tumor. And those CD8 T cells actually were expressing a reasonable amount of FOXP3 and CD25. So they looked almost more like a Treg population, but they were CD8 T cells. And we only ever saw this phenotype in mice where CD4 T cells have been depleted. And so just keep that in mind. I'm going to show you some more transcriptional data from the CD4 T cells in a minute. But so our question was, well, are these CD4 T cells maybe because class 1 is gone, maybe there's some residual class 2 throughout the tumor, and the CD4 T cells are actually responding like they would to, in a pathogenic response in some way. And so we looked at their T cell receptor landscape, and this is work from Tay Wilson in the lab. And we actually do not see, you can kind of see there's no really clean clusters here of a clear motif of T cell receptors. And indeed, if we look at the clonality of the T cells in the tumor, they're basically all unique clones. So there's no similar clones, there's no expanded clones. So there's no evidence for antigen-specific responses among the CD4 T cells in the tumor or for strong motif clusters. So it seems that the CD4 T cells are acting in an antigen-independent manner. And when we examine a little bit more the CD4 T cell clusters and transcriptionally what they're doing, this is the CD4 clusters here. There are really 3 subclusters. There were, so cluster 0, cluster 1, and cluster 14. This is work from Katie Allen in the lab. This cluster 0 looked somewhat like more conventional T cells, not super activated, but like a normal CD4 T cell, resting CD4 T cell. But these clusters 1 and 14 were really quite unique. So cluster 1 had a number of upregulation of genes, these top 3 genes that are associated with a lot of bulk RNA-seq tumor profiles, a lot of metastatic tumor profiles, and even some neurological profiles. And then this cluster 14 gets to this unconventional T cell population that I was talking about. That includes the Tregs, and there is some FOXP3, and that's here, but it's not all FOXP3 Treg cells. They look more like these other unconventional lineages like IELs, which express high amounts of Helios and CTLA4, as do Tregs at times, but they also express FOXP3. And a number of other markers of these tissue-specific, tissue-generating lineages of T cells that are more associated with wound repair, tissue recovery, and tissue development. And then they also had this very strong residency signature, which fits with the fact that they were not in the blood, they were in the tumor parenchyma. So they have these genes that move them into the tissue and can maintain them in the tissue. And this is particularly true of this cluster 14, which is this regulatory unconventional population that looks almost like an IEL or a T follicular helper cell for those of you that are a T cell subsetter. Okay, so what else was near the T cells? And so we went through all of the different cell types in the tumor. And the only cell type that significantly overlapped that we found with CD4 T cells were macrophages. And so this is also a really stark contrast to the B cells and the CD8 T cells that I showed you in the previous slides, which were all border cells. Macrophages were innervated throughout the tumor and statistically not associated with B cell or CD8 T cell localization. So B cells were actually more likely to be in areas of no macrophages than with macrophages. Whereas CD4 T cells were much more likely to be in areas with macrophages than without macrophages. And so in the last couple slides here, I'm just going to tell you about what we think the macrophages are doing. So macrophages are throughout the tumor. There are multiple macrophage types. So there's these mature macrophages and then a significant portion of these inflammatory monocytes And both of these populations, particularly this population, the inflammatory monocyte population, but a large subset of the mature macrophage population, are CCR2 dependent. And so we bred our tumor mice again to the CCR2 knockout mice. And these mice, again, are protected from tumor formation. So macrophages and CD4 T cells, the two cell types we find innervating the tumor, are both necessary for tumor formation. And if we look at what these macrophages are making, one of the genes that really came up that was striking to us was arginase 1, which is a particular interest of Peter Murray's. And Leanne and Peter for years have been working on what happens when you activate CD4 T cells in the absence of high levels of arginine. And what happens, and so this is just an activation of CD4 T cells and 100% arginine or 1% arginine with and without TGF-beta, is you induce this unconventional regulatory phenotype. And so when cells are well innervated into a tissue in a nutrient-poor environment and they get some kind of activation signal, you get this more unconventional wound repair tissue formation phenotype. And if we look at the gene expression profile of these cells that are primed and low arginine, we see basically that same profile that we saw in the cluster 14 cells, or a very similar profile in those cluster 14 cells directly outside of the tumor. And if we breed our tumor mice now to mice that are lacking arginase, which is expressed in the macrophages that we're co-localizing with the CD4 T cells, we again get a tumor protective phenotype. Okay, so that is what I wanted to show you. And so what I've shown you in this last half is the novel model of MICA and ALK-mediated neuroblastoma regulated by macrophage metabolism and lymphocyte activity. Here we think the CD4 T cells are playing a pro-tumorigenic role induced into this phenotype, this regulatory tissue form phenotype by the metabolic environment controlled by the macrophages that they're co-localizing with. But these aren't tumor-specific T cells. These are tissue-resident IEL-like populations. We sort of are starting to call these neurological tissue resident because they also match the profile of a lot of T cells that you find in the developing brain, supportive of CD4 phenotypes. And CD8 T cells, and for the sake of time, I didn't show you NK cells, either have no ligands for CD8 or are not innervating the tumor. In the case of NKs, they also seem to be blocked from getting into the tumor space. And so what I want to leave you with is thinking about these two non-mutually exclusive ways that the immune response interacts with the tumor. There's still quite a lot of therapeutic opportunities for destroying tumors with the organized response of the immune system with specificity, functional capacity, and exquisite tumor targeting. But what I think is really understudied, either theoretically or from a therapeutic perspective, is how the immune response and immune cells can coordinate this destructive organization, actually tumor growth, immune-regulated tissue formation, which they're critical in epithelial tissues in normal development. They build stromal barriers to immune control and immune tolerance and potentially contribute strongly to tumor structure. And with that, I'll close. I think I've acknowledged everyone as I went along that has contributed to the work in the lab. And I'd like to thank our funding sources. And again, thank you all for giving me the opportunity to speak. I'm happy to take questions.
Paul, thank you very much. That was an amazing survey of many aspects of interaction between the immune system and tumours in various contexts. And a reminder again for everyone who's got questions to put these in the chat function and address them to all panellists. Just before we get going with questions, we are at the top of the hour. Just to remind you that we will stay on. Paul's willing to take questions for a while yet, so stay with us if you'd like to engage in that discussion. And just a primer for the next couple of talks on November the 12th, Michelle Mongi from Stanford is speaking on targeting neuron-glial interactions in childhood brain cancer. And on November the 19th, Eric Chow from the Hutch is speaking on emerging late effects of targeted molecular therapies. So with that, some questions. The first one is from Paul Gallagher. Can you speculate as to why you're finding so many tumour-infiltrating immune cells in your mouse, which seems to contradict the recent single-cell RNA-seq papers in humans?
Sure, that's a great question, and I think it's. I think I just didn't do a very good job of explaining what I was depicting. So that was not the TC or the mu map that I was showing was not representative of just taking a piece of the tumor and grinding it up and running it through transcriptomics. We actually took the tumor, isolated the immune cells by flow cytometry sorting separately from the tumor cells, and then mixed them back together. Because if you just took the tumor and ground it up and put it through, you'd have a much higher ratio of tumor cells to immune cells. there are, in a ratio basis, there's very few immune cells relative to tumor cells, but we would argue that the immune cells are incredibly potent. But I do think you can kind of get a sense of that when you look at the spatial data, where it looked like we had all of these, B cells, if you just count cells in the tumor. But when you actually look at where they are in the tumor, they're in very isolated areas, and relative to the number of tumor cells, they're really very small. The CD4 T cells also, there's not a huge number of them, but they're actually kind of evenly spaced throughout all of the tumors.
Okay, thanks. Next question is from Stacey Ogden. So are the macrophages that are altering metabolism sending a signal to the tumor cells to change their metabolism, or are they providing specific metabolites directly to the tumor cells?
I think it's a great question. I think, the trouble with a lot of our ability to do these studies is we get caught in this chicken egg problem, where if we, so what came first? Is it the tumor somehow generating a nice environment for some of these immune cells to come in, and then that sort of a feed-forward loop to prevent any immune destruction? Or is it the immune cells actually are required to generate the tumor, which we think we at least have some evidence for now? And then there's other feed-forward loops that get set up there with potentially immune cells helping the tumor cells in terms of creating certain microenvironments. The trouble is that when we use sort of a blunt tool like a knockout, as I showed you throughout the talk, we frequently don't get a tumor. And so we can't ask what it would be like without that pathway, because there's no tumor to really interrogate. And so I think it's entirely likely that the macrophages are both creating a certain metabolic environment that favors tumor cell formation. And so there's the feed-forward circuit that way. And then we do know that the breakdown products of a lot of these enzyme pathways, I only showed you arginase here, but we've also done work on IDO and IL-4, IL-1, generate these, I always pronounce this word wrong, but kynurines and other metabolites that are either helpful to the tumor cell or required by the tumor cell. And so I think it can go both directions.
Okay, great. Thank you. So next question from Mehdi Chaib. Apologies if I've not pronounced your name correctly. In your arginase knockout model, do you think the protective effect is specific to macrophages lacking arginase, or do you think there's a contribution of arginase lacking neutrophils and immature monocytes?
It's a great question. On the neutrophils, we did have a population of cells in there that looked relatively neutrophil-like, but it was reasonably small relative to even the other macrophage populations, which weren't there in huge abundance. And then in terms of the immature monocytes, the characterization of the immature monocytes from mature monocytes to mature macrophages is a bit controversial in the field. You find different people characterizing them differently, and using surface staining versus gene expression can give you kind of different answers there. And so I've tried to be a little bit agnostic to what specific subset it's in, rather than just saying it's in a monocytic lineage. Using surface staining and other methods, we don't see a huge role for the more granulocytic neutrophil types. And so I think that that's not probably a major contributor, but among the monocyte populations, I'm not entirely sure. And I should say, just because it is such a huge topic in the monocyte field, we did look at the kind of conventional markers of MDSCs, and we don't see a really sort of typical MDSC population here, but it doesn't mean that the MDSC type activities aren't playing a role.
Okay, great. Perhaps if I can ask a question, Paul, coming back to the first part of your talk, given this kind of dichotomy between what you've shown, the high frequency of potentially autoreactive T cells in the bone marrow, at least, of leukemia samples, but at least in general, the relatively poor response of pediatric tumours to checkpoint blockade therapy. What do you think is the best approach or potential approach for switching on the anti-tumour efficacy or activity of autologous T cells?
Great question. So I think really there's two approaches. So one is to continue down the cell-based therapy type approach, where we use the endogenous T cell responses to inform us about what targets we should be going after. For instance, the fusion targets potentially seem like a good possibility to develop a CAR-like approach, where you put a T cell, TCR-T approach, where you put that T cell or have it ready to go, and you're delivered to the patient, and you're essentially then raising the precursor frequency of those cells, and you could prime the cells with cytokines and other things to raise their functional capability as well. Alternatively, I think what part of what I try to emphasize through this talk and through some of our recent papers on these topics is I think we've gotten really locked into T cell exhaustion as the end of all T cell trajectories in tumors, and it's clearly a critical one, and it's a really important one, and I think in solid tumor settings, particularly slow growing solid tumor settings, maybe the most important one. But it's not the only outcome you can have from a T cell activation event, and I think we're as much as immunologists are annoying in terms of how much we subset things, I think we need to maybe subset these things a little further and understand the nuances of these trajectories a bit more. And that might give us something akin to checkpoint blockade, but targeting these other dysfunctional cell types. If they are truly dysfunctional cell types, that, can we get the endogenous response levels at that magnitude to have an efficacy that is actually able to control the tumor? I guess one thing I fear, and I don't know if this is reasonable, but it's just sort of more of an intuition, is that particularly for something like ALL, which is another lymphocyte that can grow tremendously rapidly, that it might just be a magnitude problem where it's just hard to generate a large enough endogenous response to keep up with that tumor. So it may not be pure dysfunction, but if there is a dysfunctional pathway, I think we've just got to do more functional work to figure out what it is and figure out if there's, an antibody or a small molecule or something that can reverse it.
Okay, so from Martine Roussel, can you tell us something about the tumors that arise when you deplete myeloid cells? Would CSF1R inhibitors be helpful to suppress tumor formation and mimic deletion of myeloid cells?
I would love to look at that, because I think the CSF1 inhibitors would potentially be much more potent than what we've done even with the CCR2 knockout and some of the other approaches. We actually were just talking about this with Peter recently about reaching out to you to talk about that. As far as, the tumors that we do get in the CCR2 knockout mice, we haven't characterized those as thoroughly as we should. And that would be interesting, because we do get some. It's not, it's not like the RAG where there's literally none, but they are quite reduced. One of the reasons we haven't gone after that as much is because we know CCR2 is not, not eliminating all of the myeloid lineages that can show up in the tumor, particularly not getting rid of any tissue macrophages that could have been local and formed, that it's just not a very clean knockout, and that we'd find essentially the same sort of tissue macrophage phenotype, but we haven't looked at that rigorously.
Okay, well, I don't see more questions. So, Paul, thanks again. A big virtual round of applause for a fantastic talk. And thanks, everybody, for attending. And we look forward to you joining us again next week. Thanks all.
Thanks, everybody.